Hello friends, welcome back. In continuation of our previous video for CUCET 2021 Life Science paper, we bring you a new video where we are discussing the part B, first 45 questions. So let's begin. Which of the following electronic transition is not possible in UV visible region? N to pi star, sigma to sigma star, pi to pi star, or none of these. As we know that UV, which starts from 190 to 380 nanometer, while visible, which starts from 380 to 750 nanometer, they have got sufficient energy to promote electronic transition from ground state to excited state. Pi to pi star and N to pi star, these are the low energy required electronic transition, and they can enough be promoted by the UV visible range. While sigma to sigma star transition require high energy for electronic transition, and they are the outside the scope of UV visible range. Hence, the option B should be the correct answer. Resolving power of a microscope mainly depends on numerical aperture, refractive index, objective lens, or ocular lens. As we know that the resolving power is the ability to distinguish two closely situated points as two distinct ones. If we see the equation, that we will find that the point of resolution or the limit of resolution can be related with the lambda and numerical aperture. Generally, the resolving power is directly related to the wavelength, while inversely related to the numerical aperture. So here in the option numerical aperture is being given, so the option A would be the correct answer, that resolving power of a microscope generally depends on the numerical aperture in an inverse relationship. Which one of the following is not true according to Kasha's rule about fluorescence? Bathochromic shift is observed in emission relative to absorption. Hypsochromic shift is observed in absorption relative to emission. Bathochromic shift is observed in absorption relative to emission, or none of these. Before we understand Kasha's rule about fluorescence, we should know what is bathochromic and hypsochromic shifts. If your emitting wavelength after excitation is being shifted to higher wavelength, then it is called as red shift or bathochromic shift. While if this emitting wavelength is shifted to the lower wavelength, then it is called as hypsochromic shift or the blue shifts. Now, as a general rule, in fluorescence, when fluorophore is excited at one particular wavelength, then it goes to the excited state, and upon returning to the ground state, it definitely loses certain energy. Generally, after losing its energy, the wavelength which is being used as an emitting wavelength, that has a higher wavelength. That means what? The excitation wavelength will be shifted to the higher wavelength after emissions, and that's where it has to be the bathochromic shifts. So if you see it in the option C, bathochromic shift is observed in absorption relative to emission. That is wrong. Basically, emission is being shifted to the higher wavelength. That means bathochromic shift is observed in the emission rather than absorption. So option C is the correct answer. In reverse phase liquid chromatography, stationary phase is polar, non-polar, hydrophilic, or ionic. As we know that CUET, it invented chromatography where he used cellulose paper as an stationary phase and hexane as an mobile phase. Since hexane is a non-polar while cellulose, which is a polar, if we use such kind of a system, then we are into the normal phase chromatography. That means stationary phase is polar while mobile phase is non-polar. If we reverse the process, that means stationary phase is non-polar while mobile phase is polar, then we are using the reverse phase chromatography. So in the reverse phase liquid chromatography, stationary phase has to be non-polar, that is option B. Diethyl aminoethyl DEAE cellulose is an anion exchanger. It will prefer to bind with proteins having no charge, positively charged, negatively charged, or binding does not relate to the charge. DEAE, since it is having amino group, so it is a positively charged resins which can bind to the negatively charged amino acids or protein. So option C has to be correct. Increase in the half cell potential, that is E naught, decreases the oxidizing ability, increases the reducing ability, increases the oxidizing ability, or increases the reaction spontaneity. As we know, the ability of the substance to make other substance to lose its electron is called oxidizing power. With an increase in the standard electrode potential, the oxidizing power of a substance increases. So option C has to be the correct answer. Lowest yield of ATP is achieved in fermentation, anaerobic respiration, aerobic respiration, or both A and B. Generally, aerobic respiration yields you the highest amount of ATP, that is around 36 to 38 ATP after complete electron transport chain. Anaerobic respiration, which does not involve oxygen as a final electron acceptor, rather they use sulfate, nitrate, or even carbon dioxide as a final electron acceptor and may continue up to electron transport chain. And they can yield you a net gain of 2 to 36 ATP, 2 in the case if they are not continuing with the Krebs cycles and electron transport chain. Fermentation, which always stops after glycolysis and never continue till electron transport chain, can yield you a net gain of 2 ATP. Hence, the lowest yield of ATP can be observed only in fermentation. Which of the following is a molecular motor? Kinesin, kinetin, keratin, or dinectin? Kinesin, dynein, and myosin. These are the example of three cytoskeletal motors where kinesin moves cargo inside cells away from the nucleus along microtubules. So option A has to be the correct answer. The key driving force for membrane transport is membrane thickness, distance, molecular size, or concentration gradient. That's very easy. The key driving force for the membrane transport is concentration gradient. If it is not being facilitated by a transporter, then it is an example of normal diffusion where the molecules are being transported, generally hydrophobic nature. They are transported across the plasma membrane from higher concentration gradient to the lower concentration gradient. If we are talking about some polar molecules or hydrophilic molecules which need to pass the cell membrane, then they might be facilitated by certain transporter from the highest concentration gradient to the lower concentration gradient, downhill the concentration gradient. And such kind of a diffusion is known as facilitated diffusion. So here option D has to be correct answer, that the key driving force for membrane transport is always be concentration gradient. Sedimentation rate of a particle will be zero when density of the particle is less than the surrounding medium, density of the particle is more than the surrounding medium, density of the particle and the surrounding medium are equal, density of the particle is twice as the surrounding medium. As we know, in this question, it is being asked that the rate by which a particle is being drowning will be zero. That means the particle is no longer is being sedimenting. It will be when the density of the particle and the surrounding medium will be equal, then a particle will be in a kind of a hover situation. It is no longer will be drowning. If the density of the particle is less than the surrounding medium, then it will always be floating. If the density of the particle is more than the surrounding medium, that means it is a heavier than the surrounding medium, so it will always sediment. So here the option C has to be the correct answer, that whenever the density of the particle and the surrounding medium are equal, then it will always be in a hovering condition. This is the same concept which we used in the isopycnic type of gradient centrifugation. So option C has to be the correct answer. 70S ribosomes is made up of 50S and 30S subunit, 50S and 40S subunit, 60S and 30S subunit, 60S and 40S subunit. A prokaryotic ribosomes, which is 70S, is made up of two subunits, that is large 50S subunit and a smaller 30S subunit, where each subunit consists of one or more ribosomal or rRNA molecules with specific protein. So here the option A has to be the correct answer. For each ATP molecule breakdown, the sodium potassium pump causes movement of more sodium out and less potassium in, potassium out and sodium in for 1 basis, sodium out and potassium in on a 1-4-1 basis, potassium and sodium in the same direction. If you see it in the diagram, the sodium potassium pumps moves 3 sodium out and 2 potassium in for each ATP hydrolysis. So it moves more sodium out and less potassium in, which is option A. So option A is the correct answer. The number of a nuclear pore in a cell depends on the size of the cell, transcriptional activity of the cell, DNA content of the cell, or none of these. Nuclear pore is a point of communication between cytoplasm and nuclear compartment. When transcription is necessary, then these pores exist in large number during the cell cycle. For example, in a typical mammalian cell, generally we observe that the number of these nuclear pores they gets in double during the G1 and G2 phase of a cell division. So the number of nuclear pores in a cell depends on the transcriptional activity of the cell. Option B. Which of the following is not a part of endomembrane system? Golgi body, lysosomes, ribosomes, or vesicles. Endomembrane system include nuclear membrane, ER, Golgi apparatus, lysosomes, vesicle, or endosomes. Ribosomes definitely are not the part of these endomembrane system. So option C has to be the correct answer. Michaelis-Menten constant, that is Km, is related to the enzyme's affinity for the substrate, enzyme's activity for the substrate, enzyme's specificity for the substrate, or enzyme selectivity for the substrate. Km or Michaelis constant can indirectly be related to the enzyme's affinity for its substrate molecule. Generally, it has been seen that natural substrate they do have the lowest Km value for an enzyme. However, this could be misleading as well, because it has been seen that certain inhibitors, which are also known as suicidal inhibitor or tight binding inhibitors, their Km value is far less than the Km value for a natural substrate for an enzyme. So here we can say that option A is close to correct among the given options. The organelle absent in a plant cells is vacuole, lysosomes, chloroplast, or centriole. Centrioles are the organelle which are generally absent in the plant cells, and they do not use centrioles during cell division. So option D has to be the correct answer. Which of the following is not associated with the ECM of the cells? Proteoglycans, cellulose, collagen, or fibronectin. If you see in the figure, ECM is primarily composed of proteins like collagen, non-collagenous proteins like fibronectin and elastins, while a combination of carbohydrate and a proteins shaft, that is proteoglycans. On the contrary, cellulose is found in the plant cells while absent in the animal cells. So option B has to be the correct answer. The enzyme responsible for the removal of DNA supercoils is helicase, topoisomerase, DNA ligase, or DNA polymerase. Topoisomerases are the enzymes which are responsible for removing supercoils in the DNA helix, and they do it by transiently cleaving one or both the strands of a DNA. If one of the strands is being cleaved, then it will be done by the topoisomerase 1, while if the both the strands are being cleaved, then it will be done by the topo 2, or also known as DNA guidase. So topoisomerase are the enzymes which are responsible for removing the DNA supercoils. Which of the following is not a secondary messenger? Calcium, diacylglycerol or DEAE, cyclic AMP or AMP. Secondary messenger are the intracellular signaling molecule which are released in the cytoplasm in response to an hormone when it binds to a receptor. Some of the common examples of secondary messengers include cyclic AMP, cyclic GMP, inositol triphosphate, diacylglycerol or DEAE, calcium ions, or even nitric oxide molecule. The option which does not fall in the category of secondary messenger would be AMP, that is option D. Intramolecular rearrangement are catalyzed by transferases, oxidoreductases, isomerases, or lyases. Isomerases are the general class of enzyme that convert a molecule to its isomers or an alternate arrangement of the structure, which is also called as intramolecular rearrangement. So option C has to be the correct answer. An enzyme without its required cofactor or prosthetic group is referred as coenzyme, holoenzyme, apoenzyme, or catalyst. Apoenzyme is the protein part of an enzyme without its cofactor or coenzyme. So option C has to be the correct answer. Gram staining is based on the presence of dash in the cell wall, phospholipids, peptidoglycan, lipid A, or glycoprotein. Gram staining is based on the amount of peptidoglycan layer present in the bacterial cell wall. Those bacteria which has the thick peptidoglycan layer will be considered as positive after gram staining process. So option B is the correct answer, that gram staining is based on the presence or an amount of the peptidoglycan layer in a bacterial cell wall. Lipopolysaccharide present in gram negative bacterial cell wall is considered as an endotoxin because of, is it lipid A, is it core polysaccharide, O side chain, or integral protein. Lipid A is a lipid component of an endotoxin responsible for the toxicity of gram-negative bacteria. If you see it in the figure, then you will find that lipid A is the innermost region of the entire toxin and has hydrophobic group attached with the carbohydrate side chains. So option A is the correct answer. Antibacterial action of penicillin could be attributed to inhibition of the enzyme transpeptidase, inhibition of the cross-linking of peptidoglycan side chain, hydrolysis of peptidoglycan, or both A and B. Penicillin and other beta-lactam based antibiotic, they inhibit the antibacterial action of a penicillin derived from the inhibition of enzyme transpeptidase, which in turn inhibit the cross-linking of peptidoglycan side chain. So option A and B both are correct. So B has to be the correct option. Severe acute respiratory syndrome coronavirus 2, that is SARS-CoV-2, is caused by a single-stranded RNA virus, double-stranded RNA virus, single-stranded DNA virus, or double-stranded DNA virus. As we know, the SARS-CoV-2 is a single-stranded RNA virus of approximately 30 kb genome size and belongs to the type 4 category of Baltimore classification. So option A has to be the correct answer. For exergonic reaction, delta G naught is negative and KEQ is more than 1. Delta G naught is positive and KEQ is more than 1. Delta G naught is negative and KEQ is less than 1. Or delta G naught is positive and KEQ is less than 1. As we all know that the exergonic reactions are generally spontaneous in nature, which do occur by releasing free energy to their surroundings. So definitely, as a logic, the product will be left with less free energy than the reactant, and overall sign of free energy change would be negative. By losing free energy, generally exergonic reaction tend to reach equilibrium, and the reactions which are having KEQ more than 1, they move spontaneously towards the product formation by releasing their free energy. So what will be the conclusion? For an exergonic reaction, the delta G naught will be negative. As you know, that product will be having less free energy than the reactant, and KEQ will be always more than 1. So option A has to be the correct answer. 2-keto 3-deoxy 6-phosphogluconate is an intermediate of pentose phosphate pathway, Antner-Dodorov pathway, Amden-Meyerhof pathway, or TCA cycle. 2-keto 3-deoxy 6-phosphogluconate is an intermediate of Antner-Dodorov pathway. So option B has to be the correct answer. Product of dissimilatory nitrate reduction is ammonia, nitrogen, nitrate, or nitric oxide. Dissimilatory nitrate reduction is in a way similar to denitrification, and in another way it is different from denitrification. If we talk about similarity, that both the reaction starts with nitrate. In the dissimilarity nitrate reduction, the nitrate is being terminally converted into ammonium ion, while in the denitrification, the nitrate is being converted into the nitrogen, which is being lost to the environment. So, dissimilarity nitrate reduction is more efficient because it conserves the nitrate into the ammonium and never allow it to release into the environment. So, option A is the correct answer. Which of the following is not a type of microtubule associated with mitotic spindle? Polar, astral, central, or kinetochore? If you see it in the figure, then you'll find out that the astral, polar, and kinetochore, these are the three main types of microtubule found during mitosis, while central is not a part of microtubule associated with mitotic spindle. Which one of the following is the correct sequence of prophase 1 stage? That is very simple. If you have to remember the sequence of the stages in prophase 1 stage, then you can use the mnemonics like lazy lebra, push, dipu, didi. Now, lazy stands for lactotin, zebra stands for zygotin, push stands for pectitin, while dipu stands for diplotin, and didi stands for dikinases. This is the simplest way by which you can arrange them and remember the correct sequence of prophase 1 stage. So option D has to be the correct answer. Histones are rich in lysine and arginine, lysine and asparagine, glycine and lysine, leucine and arginine. Histones are rich in basic or positively charged amino acids like lysine and arginine so that they can bind to the negatively charged DNA backbone. So option A has to be the correct answer. Which one of the following is not true for euchromatin? They are transcriptionally active. They are less condensed region of chromosomes. Bar body is an example of euchromatin, or all of these. Euchromatin, they are the uncoiled and transcriptionally or genetically active chromatin, while heterochromatin are firmly packed and genetically inactive chromatin. So option A and option B both are correct for euchromatin. Now, as far as bark body is concerned, it is a heterochromatin and tightly packed type of chromosomes, whereas functional X chromosome is covered by the euchromatin. So, functional X chromosome is a euchromatin, while bar body is an example of heterochromatin. So, option C is the correct answer. But here is the trick. If you see, if we say all of these, that is also not correct for the euchromatin. Think about it. Proteins responsible for structural maintenance of chromosomes are condensin, cohesin, collagen, or both A and B. Structural maintenance proteins are composed of two major proteins, cohesin and condensin. Cohesin glue up the sister chromatid together, while condensin, it helps in condensing the chromosomes. So option D has to be the correct answer, that the proteins responsible for structural maintenance of chromosomes are condensin and cohesin. Activity of protein kinase A is regulated by adenylyl cyclase, phosphodiesterase, phospholipase C, or phosphorylase kinase. Protein kinase A or pKa is a family of enzymes whose activity is dependent upon cellular levels of cyclic AMP. So option A is the correct answer because adenyl cyclase is a responsible enzyme for the converting of ATP to the cyclic AMP. So option A is the correct answer. Pectin is a polymer of glucose, glucuronic acid, galactouronic acid, or iduronate acid. Pectin is a complex polysaccharide which consists of methoxyacidified poly alpha 1,4 galactouronic acid polymer. So option C has to be the correct answer. Triple helical coil structure is a characteristic of. That is very easy. In a collagen, generally we observe that three left-handed helix, they wrapped in right-handed fashion to form a triple helix. So here the option B has to be the correct answer. Which one of the following is not true for sliding filament model? Shortening of I band and S zone with no effect on A band, shortening of I band, A band and H zone, no change in I band and H zone with shortening of A band, or shortening of I band and A band with no effect on S zone. First of all, you should know in a sarcomere what is I band, what is S zone and what is A band. If you see, when muscles are relaxed, then A band is what? The entire range of the myosin present in a sarcomere unit. S zone is what? The uncovered myosin length which is not covered with the actin. Well, what is I band? The difference between the two consecutive myosin units. So when such sarcomere unit they get contracted or muscles contraction happens, then you can see I band reduces, H zone reduces, but A band remains unchanged. So option A has to be the correct answer. Which of the following protein is associated with amphibian development? Collagen, catenin, fibrin or keratin? If you see it in the diagram, when the fertilization happens, when the sperm fuses with the egg, then transiently a cortical rotation is observed in the amphibian egg. Now there is a asymmetric accumulation of beta catenin happens, and wherever this accumulates, that side is going to form the dorsal axis of the body pattern. So catenin is associated with the amphibian development. In fact, initial amphibian development and axis formation. Which of the following is not a pattern of cell movement during gastrulation? Ingression, epiboly, invasion or invagination? If you see it in a diagram, then you will find that ingression, epiboly, invagination, involution, delamination, these are the pattern of cell movement during gastrulation, while invasion is not an example of the cell movement during gastrulation. So option C is the correct answer. NRSC stands for notochord restrictive silencer elements, neural ectodermal specification and answer, neural restrictive specification and answer, or neuron restrictive silencer elements. NRSC is an acronym used for neural restrictive silencer element, that is option D, which can be used as a gene switch to make it on and off of the transcription unit in any given system. In natural condition, NRSC binding protein, that is REST, is present in all cell types except neuronal cells. Once REST binds to the NRSC, it silences the neuron specific gene in non-neuronal tissue. While if REST is not present, which is the case of neuron, so neuron specific genes remain expressed. So option D is the correct answer. With this, we conclude this video. Please do like, comment and subscribe our channel to get the more video updates. Thank you very much.